All right, welcome everyone to today's AATN seminar at a different time. Our speaker today is Yasuaki Hiraoka, who will be talking about single cell trajectory inference based on entropic Gaussian mixture optimal transport. Floor is yours.
Okay, thank you very much. First, I'd like to thank all the organizers of AATRN for giving me a actually wonderful opportunity to talk here. Today, I'm gonna basically talk about geometric data analysis in human biology. And in particular, I'd like to present a method called scEGOT, which we recently developed for single-cell trajectory inference based on entropic Gaussian mixture optimal transport. And if you have any questions, please interact me at any time. I'm very happy to answer during the talk if you have. And all right, so first, let me briefly recall the basics, some of the basics about single-cell biology. Well, as you may know, each cell contains a DNA strand like this, and genes on such a strand will be transcribed into the so-called messenger RNA. And those RNAs will produce the proteins which have the biological functions. For instance, there are 20,000 genes in mammals, including humans, and the status of each cell is characterized by measuring the expression levels of all those genes. But in the past, we couldn't measure the expression levels of all such genes in each individual cell resolutions. We couldn't do that in the past. But recent advancement of the measurement technology now allows us to measure such gene expression levels in each individual cell resolutions. This is just one of the techniques called SC, so single cell RNA-seq data, which you may know. So this is one of the format of the single cell biology. So for instance, suppose you have N cells in your cell population, then this data, so it has an output. It is given by the D by N matrix. So this is the output. So output is given by D by N matrix. Here, D is the number of genes. So for instance, 20,000 for mammals. And N is the number of cells, usually thousands or 10,000 and so on. So this is a huge matrix. And biologists are producing such a data, massive data. And each entry, Xij, shows the expression level of ith gene in J's cell. And usually, we regard this matrix as a point cloud data in a d-dimensional space. For instance, in my talk, I will consider this matrix data as a point cloud data in d-dimensional space, meaning that each column is showing the location of the point, and our point cloud data consists of n points in the d-dimensional space. So yeah, so this is some of the basics of the single cell biology, which I will use in my talk later. And actually, this is one of the frontier of the current biology. So many biologists are now using single cell technology in their research. On the other hand, here, let me briefly go back to a history of the cell differentiation in biology. I'd like to recall or explain one of the concepts called Waddington's landscape, which is proposed in 1957. Actually, this is a very useful conceptual model to interpret or to explain the dynamics for cell differentiations. And in this model, in this conceptual model, cell differentiation is explained in such a way that first we have a ball on top of the hill like this, so which corresponds to a stem cell. So stem cell, roughly speaking, can be changed into any type of cells. Then cell differentiation is explained by a ball rolling down to several valleys, and each valley corresponds to a cell type. For instance, this corresponds to a neuron, this corresponds to an immune cell, this corresponds to germ cell, and so on. So this is Waddington's landscape, a conceptual model. So mathematically speaking, this is just thinking cell differentiation as a gradient system in the terminology of the dynamical systems. Then, well, this was a very useful conceptual model, but the recent advancement of the quantitative biology requires more, actually more than just an interpretation or just a qualitative explanation. They want to be more quantitative. So for instance, what is the shape of Waddington's landscape in real cell differentiation? This means that, for instance, what is the domain where this function is defined? So we don't know such a question in this model. And also, what are the marker genes that shape those functions or level of the hills? And then to answer those quantitative questions, recently, time series single cell data are supposed or are expected to be a very promising player to answer to those quantitative questions. And actually, many mathematical and machine learning methods based on those time series data have been recently developed. And those are usually called the trajectory inference method. And in a broad sense, my talk is classified in this research area. And I'd like to develop a comprehensive data analysis method to study dynamics of cell differentiation using time series single cell data. So this is the background of my research today, of my talk today. And our approach to this problem, our approach is to apply optimal transport in single cell biology. So in this slide, let me briefly explain what the optimal transport is and how we use it in single cell biology. First, suppose we have time series single cell data like this. So at each discrete time, we do some measurement and we get single cell RNA sequencing data at each discrete time. So this is the data we can get. But here we should note that cells will be killed in sequencings at each time. This means that at each measurement, we pick up one cell and open it and measure the expression levels. So at that moment, that cell will be killed. This means that we cannot get cell-cell correspondence with respect to time, with respect to adjacent times. So such an information will be lost. But in order to recover or in order to reconstruct the continuous trajectory or continuous orbit, we need such an information. And actually, here is a place where the optimal transport will be used. Actually, OT can connect the most related cells between adjacent times by minimizing the transportation cost. And here is an illustration of what we are doing. Suppose you have a point cloud data, which corresponds to single cell RNA sequencing data expressed in our gene expression space at time 1 and then time 2. Then OT will provide us this kind of correspondence. This means that this cell at time 1 will be mapped to that cell at time 2, and so on. So this kind of correspondence will be obtained by using optimal transport. And more precisely speaking, or mathematically speaking, OT provides the so-called Wasserstein distance and optimal transport plan, roughly matching between the source and the target probability distributions. And in the framework of the single cell biology, we regard our point cloud data expressed in gene expression space as a probability distribution, just a discrete measure with a point support. Or you can call counting measure, well, not counting measure, but probability distribution as defined by the point cloud data. And we transport those probability distributions at time 1 to time 2 by using the optimal transport theory. So this is the basics of the OT and how we use it in single cell biology. But honestly, we are not the first group which applied OT into single cell biology. And let me review some of the previous existing works. one of the most famous research is done by Schebinger in 2019, which is called Waddington OT. I think this is really a pioneering work. Yeah, actually a pioneering work, and I really like this paper. But there are several issues to be improved. First, the method is based on point-to-point OT. So they are trying to get this kind of point-to-point correspondence. But then you can imagine that this requires high computational cost. This means that, for instance, suppose you have 10,000 cells at time 1 and 10,000 cells at time 2, then at least you need to compare 10,000 times 10,000 combinations. And yeah, it requires a lot of computational cost. This is one thing we want to reduce. And there are several topics to be mentioned, but next important issue is that this one. This method is not generative model. This means that, for instance, we cannot study the dynamics at the time in between. So suppose you have time 1 and time 2 measurement, but since this is not generative model, we don't know what is happening in between. So this is one disadvantage, and we want to somehow improve that. So more precisely speaking, we want to learn the probability distribution, what is happening at the time in between. This is what we want to do. And this is very important from the viewpoint of biology, since experiments will not be done so frequently. It takes time and it takes cost, money. So we want to get some kind of generative model. And speaking about the generative models, there are several methods listed here, but they are based on neural network. Then you can see that biological interpretability will be unclear due to the black boxes in neural network. So based on such a background, what we want to develop is I want to develop a cluster-to-cluster OT, which solves this problem as a generative model without using neural network. This is what we want to do. We want to develop. And we actually developed a method called single-cell EGOT, scEGOT. So this is a joint work with my former postdoc, now Yachimura-san. He's now in Tohoku University. And also Wan-san and Imoto-san and so on. And this is a collaboration with the strong collaboration with the biology laboratory. Yachimura-san is a mathematician, but the detail will be available from this paper published in last year or available from bioRxiv. And the purpose is to develop a method for analyzing time series single-cell data. And the mathematical method is based on Gaussian mixture model and the optimal transport. And here I'm showing the flowchart or pipeline from input to output. For input, we assume that we have time series single-cell data, not necessary to be RNA sequencing data. Any type of single-cell data is okay. So single-cell ATAC data, single-cell Hi-C data, and so on. So any type of single-cell data will be can be used. Then, under such setting, we have developed several mathematical theorems. Actually, we have worked on mathematics to develop several theorems regarding entropic, Gaussian mixture optimal transport. And based on that, we can provide 6 functions listed here, which are very useful in the single-cell analysis. So those are the output of our method. For instance, number 1, we can provide the so-called cell state graph, by which we can study the global picture or global behavior of the cell differentiation process. Number 2, we can provide a velocity, or mathematically speaking, we can provide a time-dependent vector field, which expresses the dynamics of cell differentiation. So by which we can do the dynamical system analysis to the cell differentiation process. And number 3, we can get time interpolation. This is very important to see what is happening at the time in between measurement times, since our method is generative model, we can do this kind of thing. And based on interpolation, we can draw animation to showing the continuous transitions, behaviors of the cell differentiation dynamics. And we can also provide gene regulatory network. And finally, if your biological system can be regarded as a gradient-like system, we can draw a landscape as a potential function. So this is what we can provide. And in my talk, in the latter part of my talk, I will apply some of those functions to some of the human biology questions. But before that, let me summarize some of the essence of the mathematical part behind this method. So using this slide and I think next slide, let me briefly explain some of the mathematics behind our method. First, for the input, I assume that we have a time series point cloud data in our space Rn at each time ti, which may have possibly different size. So this is our input data. Like those black dots are showing our point cloud data at time ti and at time ti plus 1. Then, to such a point cloud data, the first thing we will do is to assign the Gaussian mixture models to each point cloud data, like this. So for instance, if you have 2 clusters, we assign 2 Gaussian distribution to each. If you have 3 clusters, we assign 3 Gaussian distribution to that. Here, N means the Gaussian distribution, and the M is the mean vector, and the sigma is the covariance matrix. And the pi is the weight respecting the density of our data, point cloud data. And the capital K here is the number of clusters at each time ti. So here we have 2, and here we have 3 for capital K. And yeah, I will briefly remark how to select capital K. This kind of thing is always important for the real data analysis. But about the selection of the capital K, we don't do anything special. Just for instance, if you know some biological pre-knowledge, we follow that pre-knowledge to select capital K. If not, we just use, for instance, some kind of information criteria like AIC and BIC. So in any case, we set some cluster number K following biological pre-knowledge or using information criteria. Then, to such a setting, the problem we are actually solving is summarized here. It is called entropic Gaussian mixture optimal transport. Well, you may think this part is a bit dense in symbols, but let me briefly explain one by one. First, as I explained, I don't want to set up our optimal transport problem in the framework of the point-to-point setting in order to reduce the computational cost. So rather than that, I want to consider cluster-to-cluster OT. So for that purpose, we first assign a point for each cluster. So 2 masses and 3 masses for each clusters, respecting the weight of the cluster. And I will solve the OT problem in this setting. In that sense, our optimization space denoted by pi is given by 2 by 3 matrix, or more generally, Ki times Ki plus 1 matrix space, whose crumb sum and the row sum will be the original weight pi. So this is a space where we consider optimization. And then this part, minimization part, here we are trying to solve the most effective way to transport our clustered masses to the next masses, under the condition that our transportation cost will be minimum. Here, the transportation cost from K's cluster to the L's cluster is given by the Wasserstein distance between two Gaussian distributions. This part will be explicitly computable. So this is the first part. And the second part, as usual, we put a regularization term. So epsilon is a parameter, and the h is given by Shannon entropy. So this is what we are solving, actually. And one remark is that we have a unique solution denoted by W epsilon for each epsilon. So for each epsilon, we can find some W epsilon, which will be the solution of this problem. And the important point is that such a solution, explicit solution, is explicitly computable. This is really important to explicitly write down the algorithm of our method. So this is the problem what we are actually solving. And well, due to the time restriction, I cannot explain more details about mathematical part about this problem. But let me just briefly show important two properties, mathematical properties. First, it shows the convergence of our solution. Our solution, which was denoted by W epsilon, has a limit when we make epsilon going to 0. This is denoted by W star here. From this proposition, we can discuss the asymptotic behavior or converging property of W epsilon. This gives us some of the theoretical framework to study this problem in mathematical way. And then next one is explained by this theorem. I don't go into the detail, but the essence is that our solution W epsilon is obtained by solving the discrete problem. Discrete here means that we consider the OT in the cluster-to-cluster setting. But such a discrete solution W epsilon can be regarded as an approximation of a certain type of continuous problem, which can be regarded as more natural in biological setting. Here, continuous problem means that we can consider the similar problem in the space of Gaussian mixture probability measures, and our discrete solution can be regarded as an approximation of the solution considered in this setting. And this theorem actually plays a really important role to go back and forth between discrete problem and a continuous problem. And the discrete problem, from the viewpoint of the discrete problem, it allows us to have the low computational algorithm. On the other hand, when we consider this problem from the viewpoint of the continuous problem, we can get more natural interpretation of our solution W epsilon in the setting of the biological system. So in that sense, this proposition and this theorem will be really important. But I will not go into more detail today. Anyway, based on our explicit solution, W epsilon, we can provide 6 functions. And I will explain on just one of them, which will be used in the real application today. So let me explain cell state graph. So suppose we have a solution W epsilon, then cell state graph is obtained by just as a weighted graph, like weighted directed graph like this. And the vertices are given by the mean vectors of each Gaussian mixture model at each time. For instance, at time 0, we have only one cluster, and at next time, this cluster is split into 2 clusters, and the next time we have 4 clusters colored in green and so on. And then, the directed graph, directed edge is given by the optimal transport plan, namely W epsilon, from K to L, so K to L. So this is a cell state graph. So from this graph, we can study, or we can understand the global picture, what is going in the process of the cell differentiation. Say, starting from here, we have this path, we have this path, we have this path, and so on. And also we can draw the time-dependent vector field, but today I will not go into the detail. If I have enough time, after my talk, I will go back this point later. Okay, now 25 minutes passed. Then in the rest of my talk, I'd like to apply our method into the practical biological problem. So this is a joint work with Saito group in Ashby. Ashby is a human biology institute in Kyoto University. I'm also partially affiliating to this institute, Human Biology Institute. And Saito-san is a biologist, developmental biologist working in the germ cell area. And the data we have is time series single cell RNA-seq data from iPS cell to human primordial germ cell. I don't know whether you know this cell, iPS cell or not, but iPS cell is a very famous cells which can be regarded as a kind of artificial stem cell. So it means that the iPS cell can be changed, very roughly speaking, iPS cell can be changed into any type of cells. And they are studying the cell differentiation process starting from iPS cell to the human germ cell, PGCLC in short. But this is just a human germ cell. And we have 5 time points starting from day 0 up to day 2, measured every half day. So here I'm writing IMLC, but this is almost the same as iPS cell. So you can think day 0 is corresponding to iPS cell up to day 2. And we apply our method to those data. And here is the biological results we obtained. First, we identified the pathway and the progenitor cell population of human germ cell. And second, this is more important. The second is that we discovered some of the important genes, such as TFAP2A, NKX1.2, GATA6, and MESP1 for germ cell induction process. And some of them are really new in biology. So based on our mathematical data analysis, we found several important genes which were not known in biology. And in the rest of my talk, I will explain those biological results based on our data analysis. First, let me explain a little bit about germ cell, So this is showing an illustration starting from fertilization up to the production of sperm and the oocyte in the early development of human, human. And the time zone we are now dealing, so germ cell specification process is somewhere around here. So right after fertilization and up to a week or several days. So this is the data we are now studying. And biologically speaking, they want to increase the production ratio of germ cell starting from iPS cell. As I explained, iPS cell can be changed into any type of cells, but it is not so easy to produce germ cell starting from iPS cell. But for the purpose of therapy, or even in the sense of the basic science, they want to increase the production ratio of germ cell starting from iPS cell. This is what they want to achieve. And to such a requirement, we could succeed in increasing the production ratio of germ cell based on our mathematical data analysis. I'd like to explain that kind of result today. So this is the background. And here is the result of the cell state graph. We applied our method to the real data. So here we see one big cluster which corresponds to iPS cell, so day 0. And then next time at day 0.5, this big cluster is split into 2 clusters. And next time at day 1, we have 4 clusters and so on. So this is a cell state graph. And also it is easy to check that this is a PGCL, so germ cell population. So you can see that this is a path going to germ cell, starting from iPS cell. But not only germ cell, we also have amnion and endoderm and mesoderm cell types. And again, what they want to achieve is to increase the production ratio of germ cell starting from iPS cell. So they want to make this path wide, strong, somehow, by controlling the gene expressions, for instance. Then, by carefully looking at this cell state graph, we see that even at day 0.5, we have two clusters. One is going to germ cell, and the other is deviating from germ cell, actually going to mesoderm. So in that sense, we can say that this cluster can be regarded as a progenitor cell population going to germ cell. And then we carefully compared the gene expression levels between these two clusters. Here, x-axis corresponds to the cluster of this small cluster. X-axis corresponds to the cluster of this small one going to mesoderm, and the y-axis corresponds to this cluster going to germ cell. And each point here corresponds to a gene. Then we found that, for instance, TFAP2A, gene TFAP2A and the NKX1.2 are expressing high in y-axis, meaning this cluster, while GATA6 and the MESP1 are expressing high in x-axis, meaning this cluster going to mesoderm. Actually, the result about TFP2A is consistent with the previous report by Chen et al. They also found that TFP2A plays an important role going to in the process going to germ cell. So our result is consistent with that. But on top of that, we also found another new genes, for instance, NKX1.2 and then GATA6 and then MESP1. Those new genes are very new. And actually now here I'm showing the gene expression levels of TFAP2A and the NKX1.2 in the same space of the cell state graph. Then by changing colors of the expression levels, then we see that TFAP2A is expressing high along the path going to germ cell, which is located somewhere around here. So it is actually expressing high along this path. This is good, but this is also expressing high around these areas. This corresponds to amnion and the endoderm. In that sense, it is not so precise to characterize the path going to germ cell. On the other hand, the gene NKX1.2, which we found, is more narrowing down the path going to germ cell. So in that sense, we think that NKX1.2 is also very important, or maybe more appropriate to characterize the path going to germ cell. This is one new finding based on our mathematical data analysis. And based on this observation, we considered the original goal, how to achieve high production ratio of germ cell starting from iPS cell. And here is the result. Based on that observation, we conducted several knockout experiments. Here I'm showing just two of them. We did a knockout experiment of NKX1.2 and a knockout experiment of the GATA6. In the previous slide, I explained that NKX1.2 is expressing high along the path going to germ cell. This is what we observed. Based on this observation, if we knock out NKX1.2, it may be the case that the production ratio of the germ cell may be decreased. And actually, I'm showing the wild type experiment, meaning that we don't do anything, naive experiment. And then the bottom panel shows the knockout experiment of NKX1.2. And here I'm showing the production ratio of the germ cell. As we expected, the production ratio of the germ cell decreased actually from 28% to 12%. So this is consistent with our conjecture. And also we conducted knockout experiment of GATA6. As I explained here, GATA6 is expressing high along the path going to mesoderm, deviating from germ cell. So from this observation, it may be the case that once we knocked out GATA6, it may be the case that production ratio of the germ cell may increase. And actually, the production ratio of the germ cell increased from 6% to almost 80%. So in this way, we could contribute to the human biology project from the mathematical research. So this is one of the biological result based on our method.
Yes, so can I have a question before you go to the next?
Yeah.
How do you knock out a gene?
This kind of question should be asked to the biologist.
Okay.
I don't know how to do that. Sorry.
So you just tell them that you think you found this gene and that you think they should knock it out and they do the rest for you.
Yeah. So we did the mathematical data analysis and we got this kind of observation. Then we made them convinced.
Yeah, you convinced them.
Yeah, our observation is really useful. But I honestly speaking, this is a point what we really needed time. Actually, it took so long time to make them convinced to do the knockout experiments. At first, we couldn't, that they didn't, honestly speaking, they didn't believe our observation very well. They said that it may be possible, but it is not strong enough to do the new experiment, something like that. But we prepared many data and observations, and then finally they agreed to do that. And they somehow did a knockout experiment, which I don't know how to do that.
Okay.
Sorry.
No, I mean, I was just curious because I wouldn't know even where to start.
Okay.
Thank you.
Yeah. Well, my talk is almost done. I have only 2 slides. So at the end of my talk, let me briefly explain some of the ongoings around optimal transport. Today, I mainly talked about optimal transport for the purpose of interpolating time, right? So time interpolation was done by using OT. But well, mathematically speaking, we don't need to restrict our attention to only time, but we can also consider space interpolation or both. And actually, one of my postdoc, Okamoto-san, is now working on this project, spatial temporal reconstruction of gene expression dynamics from the single cell data during early human development with the biology laboratory, RF group in, again, Human Biology Institute of Kyoto. This project looks very interesting, which is relating to the pattern formation dynamics. And also, what is next after time and space? Well, we don't need to, we can also consider the cross-species analysis using OT by using the so-called Gromov-Wasserstein OT. Tokta-san, one of my postdocs, is now working on this project. But on this project, we have to be careful that so far, so Okamoto-san's project or today's project, all the point cloud data are sitting in the same space. For instance, if you are interested in the human, all data is in human gene expression space. But to study cross-species analysis, we need to consider at least two spaces. For instance, we consider mouse gene expression space and the human gene expression space, and we consider 2 point cloud data. One is in mouse gene expression space, the other is in human gene expression space. And we want to somehow compare between them. Mathematically speaking, such optimal transport problem can be handled, can be studied by using the so-called Gromov-Wasserstein OT. And Tokta-san is now working on that based on this idea. I hope I can share some of the progress in the near future. All right, so this is the end of my talk, and I'd like to thank all the collaborators relating to this project. And for detail, you can get the detail from this paper or bioarchive. And also, all the software is available from the GitHub. Okay, thank you very much for your attention. This is the end of my talk.
Thank you very much, Yasu, for a great talk. Let's all briefly unmute and give a show of hands for the speaker.
Thank you.
All right, thank you so much. So there are apparently no direct questions in the chat, but does anyone else from the audience have a question at this point? Otherwise, I'm going to start off with one, namely, how critical is the high dimensionality here? So my own experiences with single cell data is that it's massively high dimensional and thus also a little bit noisy. So how did you find this to be a challenge or an obstruction to some of the things? And how would you suggest this could be overcome, maybe even with some TDA notions?
Actually, this is a really important question. And there are several ways to answer. So first, high dimensionality of the single cell data is really important to be considered seriously. That part, actually, before this research, how to say? So easy answer is that we developed a method so-called record. I think in this AATRN seminar, you invited Imoto-san, Yusuke Imoto, and he gave a talk relating to that project, maybe. Well, anyway, so we developed a method called record, by which we can reduce the high dimensional specific noise using some kind of statistical techniques, and we used such a pre-processing method even in this method, scEGOT. From that pre-processing care, we can somehow handle such a high dimensional noise effect, first of all. And after that, we do the dimension reduction technique like PCA and so on. So even in our method, we don't directly handle the 2000 dimensional space, but we reduce the dimension into, say, some of the hundreds of dimensions. So this is a way we are handling the high dimensionality. Yeah.
Okay, very interesting. Thank you so much. Any additional questions for Yasu?
I have some questions. So have you compared your method to other like OT-based methods for like say single cell data, and do you think it's or have you like benchmarked the computational time? Yeah.
Sorry, I couldn't hear. So can you say again?
I'm wondering about comparison to other OT-based methods for like single cell analysis. Have you looked into other implementations or whether yours is faster? Like,
actually, we have very carefully conducted the comparisons in this paper for each functions. For instance, maybe you are interested in, actually, I prepared only one slide for the comparison, but for instance, you may know RNA velocity.
Oh, yeah.
This is very famous method implemented in the many type of research. RNA velocity can also provide us with the vector field like this. And also we can provide a velocity, which unfortunately I skipped today. But let me briefly explain what is the difference and what is the similarity. Similarity, well, we can provide this kind of vector field, even our method, and we actually applied both methods to today's data. As a global view, it looks very similar in the sense that starting from here, going this path and then going down, going here and then going down. But there are several differences. For instance, in RNA velocity, we do not have any stationary areas around here corresponding to amnion and endodome. But in cell velocity, we have stationary area around here and here. And biologically speaking, we should have stationary area around here and here. So in that sense, at least to this data, our method works better than RNA velocity. But I don't say that always. It is always the case. I don't say that. And also, some of the difference is that our method can apply to any type of single cell data. But the RNA velocity, they require spliced and unspliced RNA information. It means that this method is only available for RNA sequencing data. But we can apply, our method can apply to single cell Hi-C data, single cell ATAC data, and so on. This is the difference. So depending on your interest, yeah, we can, and also I should be fair. Sorry, I should be fair that our method requires time series data, but RNA velocity does not need time series data. Only single point of time is okay. So in that sense, there are several pro and con, advantage and disadvantage. Yeah, this is brief explanation.
Great, thank you.
Thank you.
Thanks a lot, Katerina. Any other questions from the audience?
Could you say a brief word about how you recover the cell velocities? Not comparing it to other methods, but just what's the quick intro to how you do it?
Yeah, idea is very simple, actually. So now we have an explicit solution of the optimal transport plan, W. So roughly speaking, we know how much masses will be transported from here to here and to here, from here to here, and so on. Then based on that, we can define more explicitly, we can define the so-called transportation plan, which says that how much weight from the point from point X at time 1 to the point Y at time 2. So we can define such a transportation plan based on our solution. Then it means that in the sense of the expectation, we know we can define this kind of mapping. This means that at the point X at time 1 will be mapped in the sense of the expectation to the next point at time 2 by considering the, how to say in English, disintegration of the measure, Radon-Nikodym measure. We can define some probability measure showing the how much point will be mapped from X to Y. So we can get this kind of probability measure. So by taking the integral, we can define the mapping from X to the next point in the sense of expectation. So once we know this kind of movement information, we define velocity very naively from X to the next point in expectation. This is how we define velocity.
Very nice. Thank you.
Very good. Thank you so much. So any other questions from the audience for the recorded part of the talk?
I would actually have one question coming back to this discovery of the genes. So I mean, you start with the data. I would like to understand how you arrived at this discovery, because I mean, you have the data, then you run your analysis, and you have some graph, which I guess you color, and so from there, how do you deduce from this that there is like a new gene or a new set of genes behind? Like, how do you read that from the data?
Okay. Let me more carefully explain that. So first, so this is one of the data analysis result we obtained, and so this is an iPS cell, the starting cell, and this is a germ cell, our target cell, and the remaining 3 cell types, amnion, endoderm, and mesoderm, are the other type of cells. And then by looking at this cell state graph, we see that from here to next time, we have 2 clusters, 1 and 2. One is going to germ cell, partly going to mesoderm, but and the other is going to mesoderm. At least we have 2 clusters at next time. So then, based on this observation, we compared the gene expressions of the cells living in this cluster and then this cluster. This graph is showing that result. It means that x-axis corresponding to this cluster, and the y-axis corresponding to this cluster. And for each, say, gene, for instance, GATA6, when we fix GATA6, we can check the gene expression level living in this cluster and this cluster. And this plot is showing the location, the ratio between X and Y. And from this comparison, we found that these two genes are expressing relatively high comparing to other remaining genes. And those two are expressing high in X axis. So this is a way how we found these new genes. I hope it answers to you.
Yeah, that's perfect, and it's also like congratulations. It seems very like
Yeah, thank you very
clever
much.
and like as an like easy to read off. Like one
Yeah.
cannot one cannot assume this is noise because it's so off the diagonal.
Yeah.
Yeah.
Actually,
And you
today
can do the same thing for a form like if you have more clusters probably, right? You just
Yeah.
compare to the diagonal. Yeah.
Yeah.
Very
Today
cool.
I only applied or explained the cell state graph application, but we can also apply the vector field velocity information. And now we just now started some collaboration with Konstantin to know some kind of separatrix, existence of the separatrix, even at the IPS cell state, to know more about the dynamical behaviors of the cell differentiations. So yeah, just a comment.
Great, thank you very much.
Thank you.
Excellent. Well, thank you so much. So any other questions from the audience for the live part of the talk? If not, thank you very much, Yasu. I'm going to stop the recording and we're going to move into the offline portion of the discussion. Thank you so much, everyone, for attending.
Thank you very much.